Okay, I'm going to teach you how to look at DNA fingerprints and then analyze them. So we are on pages 24 and 25 of your packet. And so a DNA fingerprint is basically any time that you have cut up a DNA sequence and then you run it on a gel. And when I say run it on a gel, what that means is that the smaller base fragments, so you cut your DNA up using restriction enzymes. Restriction enzymes are like literal scissors to DNA. They will cut them up at different points, depending on how many of those nucleotide sequences they recognize. So you're going to cut up your DNA into different lengths, and everyone's DNA will be cut into different lengths because everyone's DNA is different. Okay, so my DNA probably will not have the same fragments that your DNA will, and that's because our DNA is not the same. So you run it on a gel through gel electrophoresis. What that means is these smaller bands will travel further because it's much easier to fit through the small space in the gel. And the larger bands will not travel much at all because they're so big they can't fit through the gel. Okay, so small ones travel more towards the end, larger ones will be more towards the well, which is just where you put the DNA in. So with DNA fingerprints, they're used a lot in forensics, especially with identifying criminals. Like if you've left your blood at a crime scene, how can we determine if it's that person? We're going to use a DNA fingerprint. And then also through paternity tests. So, you know that show like, he is not the father. This is what they use to figure it out. So you're kind of like doing your own little science experiments when you're analyzing these and figuring out, okay, is this the dad? Is this the mom? Did this suspect do it? All those different types of things. Okay, so let's look at the first part, the introduction. We're at this image right here. Okay. I want you to notice the standard, okay? There is something called a standard. That is just a bunch of fragments that have been run, so you know what they are. So you know that this fragment at the top is 12.4 kb. The one at the bottom, which you're expecting to be much smaller, is 0.9 kb. Okay? So that's just kind of a like a standard or a marker that you use to determine the lengths of your other fragments in your samples. Okay, so if we look at pale, we see that they have two marks, two lines. One line is at 12.4 kb, and one line is a little bit smaller than 6.2. How do I know that? You look here. This line matches right here with this one, that's 12.4 kb. Then they also have another line down here. It does not exactly match up with 6.2 kb. It's definitely not 4.8. So I'd say it's probably around 6, okay? It's smaller than 6.2, but larger than 4.8. If we look at Jack, he also has two bands. One band is at 12.4 kb. The second band would be around 3 kb. It's a little bit bigger than 3. So you could say like 3.1, 3.2. And then Jill has two bands as well. One is at 12.4 kb and the other one is at 4.8 kb. Okay? So do you notice how all of their bands are different? And sometimes they might not even have the same number of bands, and that's just because everyone's DNA is different. So this is how we can eliminate suspects and say this was your DNA found at the crime scene, or how we can say you are the parent of this child, if you have a known parent of the child. So this is the type of thing we're going to look at when we are identifying suspects. Now, when you're identifying suspects, you need an exact match, okay? They have to match every single band, because you want to make sure that the suspect is the person who did it. And if they're the person who did it, their DNA should exactly match the DNA found at the crime scene. So if we're looking at these, we have the crime scene, so whatever substance we found at the crime scene, suspect 1, suspect 2, and suspect 3. Again, if you are looking for who did it, whose blood is this, it's got to be an exact match. So that means that suspect 2 is the one who did it, because you see how if there's a band right here, there's a band right here for suspect 2. Band right here for suspect 2. Band here, you have bands here, but suspect 3 didn't have a band up here, so they're eliminated. Suspect 1 didn't have a band up here where the crime scene did, so they're eliminated. And suspect 2 is the person who did it. Okay? When determining paternity, which is just like who the father is, or maternity, who the mother is, it's not going to be an exact match because you get half your DNA from your mom, half your DNA from your dad. So the bands in the kids must match either the mom or the dad. If they have bands that match neither the mom or the dad, something's not right. One of those is not their biological parent. So we have mom, dad, 4 kids. What you're going to do is you're going to see if Susie has a band, is it from mom, is it from dad? If she has a band from neither, that says that one of them is not the parent, or both of them aren't the parent. Okay? So let's go through Susie's. Move this so I can see. Okay, so Susie's first band would be right here. We see that mom has a band right there as well, so that doesn't really tell us that the dad's eliminated yet. Susie's second band is right here. The dad has a band right there, so we would say that that's a match. Notice that the mom has bands right here. Susie doesn't have any of them. That's okay. Okay, you've just got to make sure that the kid, every single one of their bands matches with a parent. Okay, so Susie has a band right here. That one's from the dad, so that one's good. And she has one last band right here, which comes from the mom. So we can say with certainty that Susie is the offspring of these 2 parents. All of her bands were accounted for. Okay, then we're going to go to Anne. Anne has a band right here. If you notice, neither parent has a band right here. So that's telling us at least one of these is not Anne's parent. Okay, and second band's right here. Mom has a band right there, so that one is accounted for. And third band right here. If you notice, they are not the same. They are not the same. So that's not from the dad. Anne's fourth band is right here. Mom has a band right there, so that one's accounted for. And then her fifth band is right here. Mom has a band right there. Okay? So we can say that Anne is her child, but is not the dad's child because she does not have any of those bands from the dad, and she has bands that have been unaccounted for. No one has a band right here. No one has a band right here, telling you that there is another father. You go to Bob. Bob's first band's right here. If you go over, it's from the dad. Second band's right here. If you go over, it does match the mom's. Third band right here. Oop, that's from the dad. And fourth band, that's from the mom. So Bob would be the child of both of these parents. And then lastly, you have Max. Max's first band's right here. The dad's band does not match it, and the mom's band does not match it. So that is neither parents. His second band, if you go over, dad does not have it, mom does not have it. Third band, dad does not have it, mom does not have it. Fourth band, dad does not have it, mom does not have it. So Max is neither the child of either of these two. Okay, one more time. Susie is the child of both. Anne is only the child of the mother. Bob is the child of both, and Max is the child of neither. Okay? So that's how you read it. If you'll flip over to the back, which is page 25, we're going to go through the scenarios, and I'm just going to tell you the answers. You can check how I did it on the keys if you're still struggling, which will be under the key folder. Miss Smith has a baby named Jessica. She believes one of the two men can be the father of her child. A paternity test is done. The results are shown below. Okay, with paternity tests, you do not want exact matches. That you have to make sure that the child's band comes from either the mom or the dad. If like none of the bands come from the mom, that's saying the mom isn't the mother. If none of the bands come from the dad, that's saying the dad is not the father. So we're right here. If you look at baby Jessica, her first band, this is the known parent. So this we know is her parent. We're trying to figure out which of these two are also her parent. So baby Jessica's right here. She got this band from her mom. Second band. Baby Jessica got this one from male 2. Okay? You see how she got this band from male 2? Third band, she got this one from mom. Fourth band, well, she could have gotten it from 1 or 2. So that could be both. This band, just from 2. This band she got from mom. This band, only 2 has it, and this band from mom. So from this, we can tell that male 2 is the father of the child. Okay? Scenario 2 is a murder scene. We found some DNA. When you're just matching DNA, it has to be an exact match. So which of these gives you the exact match to the felon DNA that was discovered? Suspect 2 would be the exact match. Scenario 3, you just have to figure out what are those sample sizes. And remember, you use your marker as your starting point. Okay, so if you have this band right here, you know that they go from larger to smaller, so that this band would have to be bigger than 3,456 base pairs. And you know that because it's up here, so that means it's larger than this number. These two are the same as this number, and you're just going to keep using the markers, okay, to answer the question. So how many base pairs is this one? You know it's over 3,456. And you would just do that for all of them. And then lastly, scenario 4, I do want you to try your best. Sometimes it's easier to color code if that does help you. I want you to try to do 4 on your own, and then obviously just check your key to see if you got it right. But I do want you to see if you can do it on your own. Okay, have a good day.